Okay, hello everyone. Thank you for tuning in to this week's Open Box Science seminar. Today we have two fantastic papers that'll be presented. The first will be on a novel mode of DC maturation that contributes directly to peripheral immune tolerance. Our second paper will be on a novel mechanism of dendritic cell migration via lymphatics. So our first speaker is Xiangyue Zhang. Um, she studied clinical medicine in China and obtained her master's degree in immunology from Peking Union Medical College and the Chinese Academy of Medical Sciences. She then moved to Germany to do her PhD at Hanover Medical School in the lab of Reinhold Forster, a legend in the field of immune cell trafficking. As a PhD student, Zhang's work focused on how APCs participate in peripheral immune tolerance. Specifically, she described the process by which neonatal stromal cells in the lymph node instruct or lead to or induce a specific subset of regulatory macrophages. For her postdoc, she then moved to Stanford to join the lab of Edgar Engelmann and to continue her training in APCs and how they contribute to immune tolerance. One of her major contributions, which we'll share today, is describing how erythropoietin, or EPO for short, programs CDC1s to become tolerogenic. She's now a senior scientist leading her own team, and she's the co-founder of ImmunoEdge, which is a company started to continue developing EPO targeting immunotherapies. Xiangyue, thank you for being here, and the floor is yours.
Okay, thanks for Daniel's kind introduction. So it's my pleasure to share our work recently published on Nature. So as Daniel introduced, it's about EPO receptor on CDC1 dictate immune tolerance. Although we are describing CCR7 positive mature CDC1, but this is more like a maturation indicator or marker for the story I'm going to tell you. I'm not going to talk to you about anything about trafficking, but in peripheral lymph node, I think CCR7 positive CDC1 are very critical for initiate antigen presenting in the draining lymph node. So here is my Twitter and email address. If after my talk you have any questions or interest to communicate further, please contact with me. Okay. So as Daniel said, this is my conflict of interest. Based on my study and another study, I'm the second author in the lab. My boss founded a company called ImmunoEdge in Redwood City. So I'm the co-founder and shareholder. Okay, so when we learn immunology, we learn that the main job of immune system is to initiate effector immune responses towards pathogens or these harmful invaders. But here in another direction of immune system is called immune tolerance. So immune tolerance is the state and responsiveness of immune system to antigens that have the potential to induce immune tolerance. So in the diseases such as allergy, autoimmune, immunity, transplantation,
Sorry,
rejection.
Xiangyue, do you want to pause quickly? I think you stopped sharing.
Sorry. I don't know how to. Wait a minute, I think. So you can see now,
Okay.
right?
Yes.
Okay, sorry. So this is my second slide. So my interest is about immune tolerance. So in allergy, autoimmunity, transplantation rejection, our goal is to induce tolerance, but to dampen the dangerous or the harmful immune responses. But in cancer, viral infection, and in a more physiological relevant setting, such called fetal and maternal tolerance, we want to achieve cancer and virus infection leverage immune tolerance. So we want to break immune tolerance, but fetal and maternal tolerance is a nature format of how immune tolerance is formed during the pregnancy period time to protect the fetus to be ejected by the mother. So I believe that a better understanding of peripheral tolerance will enable the final more targeted and refined immunotherapeutic approaches for treating autoimmune diseases and enhancing vaccination responses and improve the outcomes in transplantation and cancer therapy. So as you know, Nobel Prize was given to these two distinguished scientists, Frank Burnett and Peter Medawar, because their discovery for acquired immunological tolerance. So, and last year, Nobel Prize was given to Fred Murray and Simon Sakaguchi due to their wonderful discovery in Treg cells and FOXP3, the key transcription factor defining FOXP3 Treg cells. So, however, there is a big link. So, how Treg cells could be induced and maintained by the antigen presenting cell, and how this immune tolerance is starting from the antigen sensing, uptake, and presentation, and then this tolerogenic message is passed to the adaptive immunity, especially in T cells. I think here is still big knowledge gap. So my interest is how antigen presenting cells functional state determining T cell immune tolerance. As I told you just now, so antigen will be taken up by antigen presenting cell, especially dendritic cells, and dendritic cells need to make a decision, and then they will instruct T cell to initiate effector immune responses or induce tolerance to restrained responses. I think the balance between effective immunity and tolerance is especially critical for immune system. So my interest is in dendritic cells. As you can see here, dendritic cells are the most critical and potent antigen-presenting cells to initiate naive CD4 and CD8 T cells priming differentiation. And here are many different subsets of CD4 T cells after differentiation, and many functional states and differentiation trajectory in CD8 T cell part. However, in regard with how the functional state of antigen-presenting cell, especially dendritic cells, to instruct this distinct lineage or subset of how this CD4 and CD8 priming and differentiation and their memory and their maintenance is determined, especially from the perspective of dendritic cell functional state, remains mostly unknown. So I'm interested in CDC1. As you can see here, so CDC1 is called a conventional dendritic cell, type 1 conventional dendritic cell. They have a strict lineage development trajectory. They are defined as from HSC to monocyte dendritic cell progenitor, then under the control of FLT3 ligand and its receptor, become common dendritic cell progenitor, then become pre-CDC. There is pre-CDC1 and pre-CDC2. So pre-CDC1 will be further developed into CDC1, and the pre-CDC2 will be further developed into CDC2. So CDC1, why they are so special? Because if you must know Kenneth Murphy's work, and he defines the whole lineage, almost all the lineage defining transcription factor in CDC1 development. So CDC1 will take up tumor cells, virus infected cells, these so-called cell exogenous cell-associated antigens, and then prime CD8 T cells. This is a process known as cross-presentation, particularly in vivo. So in mouse, the typical surface marker for CDC1 are XCR1, but they are alpha negative. They undergo tolerogenic maturation upon aflatosis. As I told you just now, CDC1 are specialized in taking up tumor cells and virus infected cells, and the process of taking up apoptotic cells is called aflatosis. So they will, from this cell-associated antigen sensing uptake mode to cell-associated antigen presenting mode. Previously, the main job of CDC1 was thought mostly is about cross-presentation, but 2 papers published in 2020, one from Keith Murphy's lab, one from Bernard Madison's lab, showing that CDC1 could also efficiently present cell-associated antigen to CD4 T cells. And the MDC class 2 mediated antigen presenting cells presenting process of CDC1 determines the CD8 T cell fate. This is also called CD4 T help cells licensing. So in the past decades, here are many excellent works showing that dendritic cells, after taking up apoptotic cells, they will become tolerogenic. The most recent study from Victor in Science Immunology showing that in the homeostatic state, CDC1 will undergo tolerogenic maturation, and they wrote a review in Nature Reviews in Immunology talking about homeostatic CDC1 maturation. And in 2020, Miriam Rad gave a concept called MRAG-DC. So this is in tumor citing both in human and mice. These dendritic cells, after taking up tumor antigens, they will become regulatory state. This functional state is quite like mature DC state, but later on, people also show in inflammatory setting MRAG-DC single cell RA sequencing defined functional state is there. Therefore, there is a question. So what's the mechanism underlying the tolerogenic versus the immunogenic maturation of CDC1, especially about their tolerogenic maturation? So I'm interested in from antigen sampling sentinel mode to antigen presenting mode, how this CDC1 become tolerogenic, and consequently how this will influence on their interaction, induction, maintenance of focus Treg cells, and their cross-presenting capacity and other related processes. So when we study a specific question, we need a perfect model. So TLR-ATS is a transplantation tolerance model. This was designed by Samuel Strauber in Stanford and used in clinics in Stanford clinics. So as early as 50 years ago in 1976, Samuel Strauber published a science paper showing that this total lymphoid irradiation treated mice will accept this skin allograft for the whole life. It's a lifelong tolerance inducing method. And for the next 30 years, he developed this method and used it into Stanford clinics for HSC transplantation patients and kidney transplantation patients. This is how the instrument look like to perform total lymphoid irradiation. And this is the allogenic transplantation system. The black mice will be given the treatment and then will be infused with the white mice bone marrow. And after here is a stable bone marrow chimerism establishment, the solid organ will be transplanted. And we will see the heart will be survive in the allogenic recipient forever. Therefore, what's the interesting point in this model is this is allogenic antigen-specific lifelong tolerance model, and the bone marrow chimerism is indispensable. That means we will have a strict time window for studying how the overwhelming antigen introduction will induce this tolerance. That's the key point for when the antigen-presenting cells and the dendritic cells will perform their tolerogenic function. So we found that in beta F3 knockout mice, that is CDC1 deficient mice, the whole system will be broken down. So that means here is no stable bone marrow chimerism, and the transplanted heart will be gradually rejected. And then I did back-RA sequencing to compare the transcriptome of CDC1 before and after TLR-ATS. Interestingly, EPOR ranked as the top one upregulated gene. This is the fold change most, not the expression abundancy. Actually, EPOR expression is still very low, but we capture its upregulation by doing this comparison. And then we found that in the spleen, after irradiation, here are abundant apoptotic cells, and in the spleen, the white pap will be the red pap will be destroyed. And here are many white paph also destroyed. You can see the spaced place will be occupied by erythroprogenitors. And in the same line, here is gradual ablation of EPO in the serum. And also by using EPO-RTD tomato report mice, we found here is increased percentage of CDC1 from the normally 20 to more than 60. And here is obvious upregulation EPO-RTD tomato, especially in CDC1, but not in CDC2. And then we perform the biochar sequencing again by compare this EPOR positive and negative population. Again, this gene site popped out, and EPOR was still ranked as top one. And interestingly, EPOR positive population have the immunoregulatory erythropoietin active, and they are active in lipid metabolism and iron metabolism. So it's a very interesting phenomenon. Therefore, we think that this erythropoietic active CDC1 tolerogenic function may be regulated by EPO and EPOR signaling pathway. So then we knock out EPOR specifically in CDC1 by using XCR1 and again we found the whole system was broken down. And by using this 2W-VAS BABC to use a surrogate antigen, and then we can check the host antigen specific Treg cells or CD4 T cell fate by using tetramer staining. So we found that in the control mice, after bone marrow introduction, here is more than 50% of the antigen-specific CD4 T cells are Treg cells. However, when EPOR is not there on CDC1, this will be abrogated, the antigen-specific Treg induction. So what's the mechanism? So as I told you that in the spleen, by single RNA sequencing, we can separate CDC1 from pre-CDC to late mature CDC1. And this late mature CDC1 is featured by CCR7 expression and CD63 expression. This is quite similar as the MRAC-DC signature, but actually it's a common maturation gene signature. And interestingly, we found after TLATS, the early mature and late mature stage will be skewed towards late mature stage. However, in the absence of EPOR, the late mature stage with tolerogenic maturation will be interrupted, and this cannot undergo tolerogenic maturation toward the CCR7 positive mature stage. And we did flow cytometry and found very similarly, here is much less CDC1. As you can see in wild type mice, here is expansion after TLATS from 24 to 60, but without EPOR expression on CDC1, this expansion will be abrogated. And we also found several Treg induction and recruitment associated genes were controlled by EPOR expression in CDC1. And by functioning study, we found this integrin beta 8 is the most critical downstream effector under EPOR control in CDC1 for TLATS-induced tolerance. And therefore, I show you a diagram for summarize what we found here about the mechanism. So as you know, in the spleen, from pre-CDC1 to late mature CDC1, CDC1 undergo few stages. So especially in late mature CDC1, they have this phagocytotic capacity. They will take up apoptotic cells, then become early mature CDC1. Then they will go into the spleen white paph to interact with naive T cells for priming job. And this later mature CDC1 are the functional performing state of CDC1. And they have this MRAC-DC signature. And we found that actually EPO and EPR signaling pathway is controlling the tolerogenic maturation and the expansion of splenic CDC1 towards late mature tolerogenic CDC1. So So absence of EPOR on CDC1 also give rise to more immunogenic spleen CDC1 phenotype. This is in the spleen, how the CDC1 look like. As you can see here, CD40, CD80 upregulation, but here is downregulation of PD-L1. This is very interesting. This only happens in CDC1, but not CDC2. Then we did this adaptive transfer to use apoptotic acting membrane over thymocyte as antigens and did this cogenic OT2, OT1 and OT2 injection and track their fate by cell trace violet staining. And as you can see here, in the absence of EPOR, here is a better cross presentation and a better proliferation of the OT1. And here is also a better priming and proliferation of OT2. That means CDC1 expression of EPOR limits both CD8 and CD4 T cell priming against these cell-associated antigens. So as I was trained by Reinhard Forster, so I was very sensitive about the knowledge point in peripheral tissue. CDC1 will undergo maturation and upregulate CCR7, and through afferent lymphatics, they will drain into the draining lymph node and to take the job to initially prime naive T cells. And pre-CDC1 also could undergo these vessels, and within the lymph nodes, they will become this resident CDC1. Actually, for the later stage CD4 and CD8 immune response, there is a cooperation between migratory CDC1 and resident CDC1 because the lifespan of dendritic cells is very short in the mice, is only about 8 days. So migrated CDC1 are matured CCR7 positive CDC1 originated from peripheral side, and they are critical in priming naive CD8 T cells in draining lymph nodes. And Ralph Steinman proposed long time ago this induction of tolerance by dendritic cells after taking up apoptotic cells is very crucial, and many papers showing in the past decades migrated CDC1 seems like are specific in inducing Treg cells in the peripheral lymph node. So my question is, how about EPOR expression in CCR7 migratory CDC1? So by taking advantage of ZBTB46 to easily define conventional dendritic cells, I found that in dendritic cells in the peripheral lymph node, here is very obvious small population of EPOR positive population, and most of them are CDC1, but they only mostly reside in migratory compartment, not resident compartment. By playing around with the gating strategy, we found that XCR1 CDC1, migratory CDC1, have more than 30% of expression of EPOR, but not in the resident compartment. And by utilizing different knockout model, we confirm this is a CDC1 specific and CCR7 dependent expression pattern. But this doesn't matter which peripheral lymph node it is, because in either inguinal, axillary, branchial, or cervical lymph node, here is quite similar expression of EPOR TD tomato. So how about the function? So we checked in mammary fat pad, the CDC1 express much higher percentage of EPOR, more than 70%, and then we inject this green color labeled apoptotic cells to track in the draining lymph node which kind of cells take up them. We found migratory dendritic cell takes them more than resident dendritic cells, and migratory dendritic cells after the migratory dendritic cells taking up this peripheral derived antigens, they also have comparatively higher expression of EPOR. And then we introduced the apoptotic acting membrane over thymocyte, and by giving additional EPO, we found the EPO can enhance the peripheral Treg induction capacity of CDC1 in peripheral lymph node, but this is abrogated when EPOR is absent, is deficient. So EPOR. So we also found that in peripheral lymphocyte, here's much less migratory CDC1, and meanwhile they express lower expression level of XCR1. So this is consistent with in spleen the CCR7 positive CDC1 frequency, and their expression of XCR1 is also much lower. And here is higher CD40 expression in peripheral lymph node migratory CDC1. And we did Bakari sequencing to compare or to try to understand whether in different anatomic sites, such as in spleen and peripheral lymph node, whether in CCR7 positive CDC1 or migratory CDC1. So this transcriptome was controlled both by EPOR expression with quite a similar mechanism. The answer is yes. So EPR is controlling the upregulation and downregulation of more than 300 genes, quite similarly in both sides. With the loss of EPOR, here is a better MRC class 2 mediate antigen presentation and MRC class 1 mediate cross presentation. Here is better co-stimulation, but here is lower expression of PD-L1. So CDC1, as you know, is the most critical dendritic subsite in anti-tumor immunity throughout the entire cancer immunity cycle. So this cancer immunity cycle was introduced by Aaron and Daniel Chen, Thomas and Shannon Turley, and this is most famous in 2023. They published the initial review actually in 2013. So CDC1 is are not only important in tumor draining lymph node because their cross-presenting capacity to prime naive CD8 T cells, but they are also critical in the tumor site for reactivate the TPX cells and determine their TPX maintenance or reservation and their differentiation into either exhausted T cells or effector T cells. So here is evidence showing in 2023 the interaction of Treg cells with CDC1 both in the draining lymph node and in the tumor site will impede their cross-presenting capacity and influence the CTL immune response quality. So therefore, because T cell exhaustion is the rate-limiting step for anti-PD-1 efficacy, and stem-like CD8 or TPX reside in CDC1 niche in tumor microenvironment, and TPX are generated by CDC1 mediated cross-priming in TDLM. My question is, if both in tumor draining lymph node and the tumor site EPOR is expressed on CDC1, how about the anti-tumor immunity if EPOR is removed from CDC1? So Daniel must know this very well. Here are serious papers showing that TDLM is the most critical site for TPX generation, and the TPX is the most important CD8 and tumor antigen-specific CD8 subsite in controlling the whole anti-tumor CD8 immune responses. So I have experimental evidence showing that both in TDLN and in tumor site CDC1 preferentially express higher level of EPOR. So after knockout of EPOR on CDC1, we found here is tumor reduction. And most interestingly, as what was hypothesized, in the tumor draining lymph node, here is a better proliferation and priming of naive CD8 T cells, OT1 cells. This is antigen specifically targeting this over carrying B16F10 tumor. So that means here are more TPX generation, but they still have this SLAM F6 expression, means they keep their TPX identity. In the tumor site, they are reserved much better if EPOR is not there. And here were much less focus rate T cells, especially this THAF1-like Treg cells. So accordingly, there is synergistic effect of anti-PD-1 with EPOR deficiency in CDC1. So therefore, to summarize, our finding revealed EPOR signaling in CDC1 serves a conserved mechanism, promote CDC1 tolerogenic maturation, and regulate cell-associated antigen-specific T cell tolerance. And therefore, we believe we establish a new paradigm for the tolerogenic function of CDC1. And we are redefining how maturation coordinates signals 1, 2, possibly also 3 along a tolerogenic axis in CDC1. So since EPO is always there, physiologically is essential for red cells production. So CDC1 maturation actually is by default controlled by EPOR signaling pathway towards a tolerogenic state, even in the homeostatic state. And because EPO and EPOR level both could be regulated systemically and locally, therefore the functional threshold of this tolerogenic axis is highly sensitive. And we also found EPOR as a molecular switch to facilitating the tolerogenic maturation while restraining the immunogenic maturation. This doesn't matter is in peripheral lymph node or in the spleen. And in the entire cancer immunity cycle, both at the site of tumor draining lymph node and in the tumor site, the EPOR signaling imposed tolerogenic mode CDC1 is restraining the effective anti-tumor T cell immunity. So here is a very important concept, T-help licensing. All these papers are from Kenneth Murphy, which means the CD4 T cells functional state will transfer through CD40 and CD40 ligand. This is most critical interaction, and then will determine how the CDC1 prime, CD8 T cells, and the CTL result. However, now we give another mode. We propose that EPO-EPOR signaling pathway in CDC1 promotes a tolerogenic mode of this three-cell triad. So that means we are adding a new layer of mechanism in talking about this CD40 have licensing to CDC1. And we found by single CRI sequencing without EPOR, actually this CD40 have signature. This is more than 200 genes. This is single cell sequencing based gene signature. Here is a better signature of CD40 have licensing. That means if we remove EPOR signaling pathway, and this mode will be skewed, but not this mode. So that means EPOR regulates CDC1 functional programming not only during maturation, but also in the three cells interaction, thereby regulating CDC1-mediated CD4 help cell licensing, which is essential for effective CD8 T cell response. So therefore, we believe our paper make a new conceptual breakthrough and a translational inflection point in CDC1 immunobiology. So if you have interest, you can scan this code and to read the paper with more details. If you have more questions, welcome to communicate with me. And this project actually was started a long time ago, as early as 2017. At that time, here was no knowledge about this. Here was no XR1 crate, no EPRTD tomato report mice. So this is a very long journey. So during this journey, I thank my mentor, Edgar Engelmann, very much. He gave me tremendous freedom to conceptually and experimentally develop this story. And even it's very long time project. And thanks for my lab member and my collaborator. Yes, they also give me a lot of help. And thanks for your attention. Welcome for any questions. Thank you.
Okay, awesome. Thank you, Xiangyue. So unfortunately, just because of time, we're not going to be able to have time for any questions.
Okay.
But folks,
No
if anyone has questions, please reach out to Dr. Zhang
Yeah.
from media.
Okay, thank you. Thank you.
Yep, no problem. All right. And so our next speaker is Victor Collado-Diaz. And Victor also has a fantastic pedigree in immunology and APC trafficking. And just quickly to say, Victor is currently a Ramon E. Cajal tenure track investigator at the University of Valencia, where his lab studies heart fibrosis and systemic vascular inflammation. Today, he'll present some of his beautiful work that he did in collaboration with his postdoc mentor, Cornelia Helen, on a novel mechanism of DC migration via lymphatics. Victor, if you want to go ahead and share your screen.
Yeah. Do you hear me?
Yep.
Yeah,
Yep.
okay, perfect. Do you see now the screen, I guess?
Yep, it's just sharing now.
Okay.
Okay.
All good now?
And click on presenter mode.
Yeah, is
Awesome.
it now? Yeah,
Looks
perfect.
beautiful, Victor. Thank you.
Okay, so thanks for the introduction and thanks also for the invitation to present here today in this open box seminar. This is the project that I started during my postdoc time at ETH under the supervision of Cornelia Hallin. And first of all, I want to thank her for all of her work, but also before starting, I would like to thank Nefelia and Philip, which are also my first co-authors of this paper, which they also were a big help during all the time working on this project. And also, if you have any questions and you also want to reach out after, you have here my email. And I would like to start by introducing the afferent lymphatics, which are very important for the transport of antigen and leukocytes and the induction of adaptive immunity. And as we all know, and we all saw, one of the main migratory leukocytes are dendritic cells, which are the main cell type which I will focus today. And this migration is upregulated during inflammation. But the afferent lymphatics are composed by two different vascular beds. And this is because first we have the initial capillaries, which they have this oak leaf shape, and they have gaps between them. So it's rather easy for the leukocytes to transmigrate inside the lymphatics through these gaps. Once they reach the inside, they crawl and they arrive to the second vascular bed, which is the lymphatic collectors. Now the lymphatic collectors, they have a different morphology. They have tight junctions and they're forming bulbs. Therefore, these collectors, they have a pumping role. And the leukocytes, once they arrive to these collectors, they rapidly flow towards the draining lymph nodes. And the main cytokine driving this dendritic cell migration is CCL21, which is constitutively expressed in the lymphatics. They are kept as intracellularly, like as granules, and upon stimulation, they are secreted and they stay expressed on the cell surface of the lymphatic capillaries. You can see here how LIFE1 is a staining for lymphatic capillaries and is completely aligned with the expression of CCL21. So the dendritic cells, upon maturation, they will upregulate CCR7, which is the ligand of the CCL21. The CCL21 is creating a gradient, so it's highly expressed around the lymphatics and is sensed by the CCR7, which let the cells migrate through the interstitium until they arrive to the lymphatic capillaries where they transmigrate. Then there is another intralymphatic gradient that is guiding these cells until they arrive to these collectors. As I said before, they will rapidly flow thanks to this pumping role, and they arrive to the subcapsular sinus of the lymph node. And now here, again, there is another gradient that leads the migration of the cells into the parenchyma, where they will initiate the immune response. So to go further and deeper into the CCL21, the CCL21 upon secretion stays bound to the heparin sulfate expressed on the helices. And this is because they present a positively charged C-terminus that can bind to the negative side of these heparin sulfates. And this is what we call immobilized CCL21. It's expressed on the cell surface. But this chemokine can be cleaved. There are some reports that they said that can be cleaved by dendritic activated T cells or even plasmin, generating what we call as a soluble CCL21, which is smaller, and now it doesn't have this elongated C-terminus that is binding to the helices, and is more chemotactic. And recent reports, they have detected both isoforms in tissues, and both can be scavenged by the ACKR4 enzyme. So when there is no AChR4, we can see how this chemokine accumulates in the tissue. And it's been shown that this accumulation of the soluble CCL21 and the immobilized CCL21 is reducing dendritic cell entry into the dermal lymphatics. But it's well known how this full length, so the one that is expressed on the lymphatics, how it acts, and it's really well established its contribution. But what we don't know is how this soluble CCL21, the one that is cleaved, impacts in vivo migration and how it's generated in tissues. And this is what we want to address in this paper published in this Journal of Cell Biology. And this is kind of spoiling our results, but this is I'm going to guide you through this conclusion, where we saw that uPA-UPAR is mediating this cleavage by helices. And we will see how the balance between the full length and the soluble can regulate dendritic cell migration, which is CCR7 dependent. So our first observations were using an inflammation model, which is CHS. It's a contact imperceptibility response. So you induce inflammation in the ear and the footpad of the mice. And when we perform IVM in the ear, we can clearly see here how in green, we can see the dendritic cells that they go closer to the lymphatics and they migrate inside, and then they are crawling inside these lymphatics. So to see how was the migration of the cells towards the draining lymph node, what we did is we adoptively transfer wild type bone marrow dendritic cells and CCR7 knockout dendritic cells, meaning that these cells cannot sense the CCL21 gradient. We injected, we labeled them, and then we injected into the footpad, and we analyzed the draining lymph node after 24 hours, which was what we saw is like upon inflammation there are more dendritic cells migrating, and that the CCR7 they don't migrate not in a steady state or inflammation, and the ratio of wild type CCR7 dendritic cells was higher upon inflammation, meaning that upon inflammation the migration is more CCR7 dependent. But so how this gradient, how this full length and soluble looks like in the ear, right? So surprisingly, what we saw is like we took the ear of the mice, and then we did a staining with the lymphatics, live one, and CCL21. In a steady state, we can clearly see how the immobilized gradient is expressed around the lymphatics. But upon inflammation, where we saw more dendritic cell migration, we see no expression of this immobilized, of this full length. So this is affecting the gradient, and this was a collaboration with David Lavender, who did this distance mask gradient analysis. And we saw how the intensity is higher in a steady state, close to the lymphatics, and the farther that we go is decreasing. So how is this even possible? Because even, as I told you, this CCL21, it can be found in stored intracellularly. So in a steady state, we can clearly see the granules of CCL21 inside the lymphatics, and upon inflammation, we barely see CCL21, meaning that this CCL21 is secreted, but it's not in the cell surface of the helices. So to explain this, we took the ear and we did a Western blot of this protein extract of the murine ear, and we always use recombinant CCL21 as a control. Okay, this will be representing the full length. It's a recombinant protein. And what we saw is like in both cases, in a steady state at CHS, we could find in the ear the full length and the soluble one. The soluble is the one that it kind of aligns with 11 kilodaltons, and the full length is the 15. And this we double check by using human recombinant protein. The full length will be this one, and the soluble will be this one. So since the small one is more soluble, this was reported. What we wanted to do is like an elution assay. In this elution assay, we read the ear, and we let them in medium for 24 hours. So the soluble protein will be elute to the medium, and this medium can be analyzed by Western blot and ELISA. And what we saw is like in the medium, we could only find the soluble one, not the recombinant. This is the control. And it was upregulated. So there was more CCL21 soluble in CHS compared to steady state. And this is also a quantification that we did with ELISA with an antibody that can detect both. So this is very important because it means that there is more cleavage upon inflammation. And more importantly, what we saw is like taking this medium from a steady state.chs, we did a chemotaxis assay, and we analyzed the number of wild-type or CCR7 knockout cells migrating in medium with from steady state ears and CHS ears. In the CHS ears, we have more soluble than in the steady state. And what we saw is like there were more wild-type cells migrating upon inflammation than in the steady state conditions, meaning that also we did a 3D collagen assay, and we observed that the CCL21, the soluble one, was more chemotactic than the full length. So now we know that upon inflammation, we have more migration and we have more cleavage of this CCL21 full length. So meaning that we have more soluble CCL21. And as I said, some reports were claiming that plasmin can cleave CCL21. And plasmin is very important for fibrinolysis, wound healing, angiogenesis, and tissue repair. Plasmin is formed by activation of plasminogen by two different enzymes, TPA, which is mostly expressed in blood vessels, and uPA, which can also be expressed in leukocytes and also fibroblasts. We wanted to see whether, okay, what is happening in the tissue, what is cleaving CCL21? It could be plasmin. So we did first an in vitro experiment where we observed that plasmin can cleave recombinant murine CCL21. Here we can see how the longer we wait, we can observe like more cleavage even at 240 minutes. Almost everything was cleaved. And this was plasmin dependent because when we use a plasmin inhibitor, this cleavage was reduced. Moreover, okay, if it's plasmin the one cleaving the CCL21, we should have plasminogen and more plasmin activity in the ear. And this we analyzed, and we saw that upon inflammation, we have more plasminogen, probably leaking from the blood vessels, and also we have more plasmin activity. And plasmin can be cleaving CCL21. And when we pre-treated the mice with an inhibitor of plasmin, we observed that this activity was completely abolished. So what we thought is like, okay, probably under inflammation, we have more, the vasculature is can leak more. So we have more plasminogen arriving into the inflamed tissue. And here plasminogen is being activated to plasmin, which is probably cleaving CCL21, but we have to prove this. And what is really cleaving the CCL21 here? Since the helices are the main CCL21 producers, it could be that the helices are the one cleaving this CCL21 and modulating the gradient. So what we did is we took helices, we isolate helices, and we did a flow cytometry, and we detected the presence of uPA, UPAR, and plasminogen on the surface of murine dermal helices, and if this expression was upregulated during inflammation. And this is very important because uPA needs its receptor that is expressed on the helices. So uPA binds to UPAR, and once it's bound, it can activate plasminogen to plasmin, and then plasmin can cleave CCL21. So far we know that these molecules are expressed here. And we kind of develop an in vitro CCL21 cleavage assay where we incubate helices in a monolayer. We added recombinant CCL21 that will be bound to the surface of the helices. And we also added plasminogen in order to boost this reaction. So if the helices, they have uPA and UPAR, they will be activating plasminogen into plasmin that can cleave CCL21, and this soluble version can be analyzed in the cell culture medium. So indeed, Also, we use some inhibitors to kind of confirm that it was plasmin mediator, the plasmin mediator. And what we saw is like when we only treat with plasminogen and CCL21, there was a cleavage of the full length into the soluble one, the truncated one. But when the helices were pretreated with a plasmin inhibitor or an anti-UPA antibody, this cleavage was delayed. So we had less cleavage of CCL21 when we treat with this plasmin inhibitor on UPA. So this will mean that the helices can activate plasminogen to plasmin, generated this soluble CCL21. But we need to confirm this. Is only UPA mediator or not? So we got two mouse lines. One is called UPA knockout. So this UPA knockout, they don't have UPA. Therefore, UPA will not bind to UPAR and cannot activate plasminogen. And another one is called UPA mute. So here UPA is mutated and cannot bind to the UPA receptor, and the same will not activate plasminogen into plasmin. And we performed the same in vitro CCL21 cleavage assay. What we saw is like the wild-type mice can cleave CCL21, but there was no cleavage in the UPA mute or the UPA knockout. So these knockout mice, they display reduced CCL21 cleavage. To confirm this, we need to analyze the ear to see how the gradient looks like here. And since we had less cleavage of this full length expressed on the lymphatics, we observe, like in both UPA knockout mice, there is more intensity of CCL21 of this immobilized gradient on the lymphatic vessels. So this is because, of course, there is less cleavage, and now there is accumulation of this full length on the lymphatics. And to confirm this, we did the same just in the wild-type mice, and wild-type treated with the plasmin inhibitor and this UPA. And we got the same result. There is CCL21 accumulation in the cell surface of the perilymphatic vessels. And here we can see how is the intensity at 0 and at 30. So there is less CCL21 soluble here in this mutant and this UPA. So we just said that the CCL21, the soluble one, is more chemotactic. So this is really affecting migration? Do we have less migration or less cells arriving to the lymphatic vessels? We took the ears of wild-type mutant and the UPA knockout, and then we performed a CD11C staining. So we were staining dendritic cells. And here in blue, we see the live one capillaries. And here in gray, what will be the collectors, which are also CD31 positive, like 1 negative. And here in yellow, we see the dendritic cells, which are CD11C and CD45 positive. So we analyzed, we quantified the dendritic cells that they were inside the vessels, the ones that they were colocalized, so meaning that these cells, they have sensed the gradient, and the ones that they are far from vessels. What we observed is that in the UPA and the UPA knockout, we had less cells around the lymphatics compared to the CCR7, and the CCR7 cannot sense the gradient. Here, the cells, they cannot sense also the gradient because there is no soluble CCL21. All the CCL21 is expressed on the lymphatics. So if they are far from the lymphatics, they cannot be guided to migrate there. So to summarize what we have seen so far is like we saw that the dendritic cell migration is enhanced in inflamed skin and is CCR7 dependent. Here is the wild-type inflamed and here the steady state. And that this perilymphatic gradient is diminished. We have more cleavage, so we have more of these blue light dots, which will represent the soluble CCL21. And this one has more chemotactic activity. This is why we have more migration towards the lymph nodes compared to the wild type. We also saw that this cleavage is happening because the helices can activate plasminogen to plasmin, and they generate this soluble version of the CCL21 with enhanced chemotactic activity. And in the case of these mice lacking UPA, what we saw is like there is less cleavage, so we have less of the soluble one, and there is more, this line of the dark blue, express more full length expressed around the lymphatics. So this means that we have less migration of cells towards the lymphatics. And now the last part, we are going to focus in the gradient that is happening in the subcapsular sinus in this lymph node. Because one really important thing is like the soluble one can drain by itself towards the afferent lymphatics and arrive and accumulate in the subcapsular sinus. So if we have more of this soluble one, it will be accumulated in the subcapsular sinus. So it might be that this can also, this gradient, this can be also modulating the entry of dendritic cells inside the lymph nodes. So what we wanted to do is like, okay, let's see how is this CCL21, the levels of the soluble and the full length in the lymph node. And what we saw is like in the lymph node, we barely have soluble CCL21 compared to the ear skin. So we were thinking, okay, maybe in the lymph node, the soluble, it doesn't enter, or maybe there is less cleavage. So in order to analyze the CCL21, which can be accumulated in the subcapsular sinus or in the connected lymphatics that is coming from the tissue, we perform an elution assay. Because now only the soluble will be detected. And what we saw is like compared to the extract of the lymph node, where we can also analyze the CCL21 in the parenchyma, in the L8, we only have soluble CCL21, and the same in serum. So we can say that the CCL21, the soluble one, is mainly present in the subcapsular sinus and the connected lymphatics in tissue fluids. This we quantified by ELISA using an antibody that can detect both, and using an antibody that only detects the full length. And in the L8, we didn't detect any full length, the same as the serum, but it was only detected in the extract. Whereas the soluble one was mostly detected in the L8. So what is happening now in the mute? The mutant, these ones that they don't have UPA, that they have less cleavage in the skin, in the tissue, so there is less soluble that can drain into the subcapsular sinus, and we can clearly see how these knockout mice, they have less concentration of the soluble one in the subcapsular sinus. And this is consistent with an observation of a previously published paper where they saw like when there is more soluble CCL21 in this ACKR4 knockout mice, there is accumulation in the subcapsular sinus. So now we wanted to explore what is happening with the levels of the CCL21 in both in this UPA. What we saw surprisingly is like we detect exactly the same intensity of the CCL21 in the subcapsular sinus, the full length, the immobilized, and the parenchyma. Here we can see the T cell zone, which represent the parenchyma, and also in the B cell zone was excluded. So it seems that the cleavage in the drainage lymph node occurs at a lower rate compared with the skin. But how we can see if this is the migration into the lymph nodes is being affected by this reshape gradient. So as I said, what we saw is like the CCL21 can drain and accumulate in the subcapsular sinus. And there are some reports saying like, if you have more of the CCL21 accumulated, now here, the dendritic cell entry is kind of impaired. So there is less entry of dendritic cells when you have more of the soluble CCL21 into the parenchyma. Because this soluble CCL21 is less sticky and it can obscure the full length. This is the one that the cells they need to enter. So it could be that in our mutant mice, we have more entry since we have less soluble. So exactly the contrary, what it was shown with ACKR4 knockout mice. So to do this, we did this is the last part. We did what is called a FITC painting. So we treat the mice with FITC, and now this FITC is a molecule that will be uptake by the dendritic cells. So now these migratory dendritic cells can be detected in the lymph nodes because they will be expressing FITC. So the ones that migrate from the ear to the auricular lymph node. And we did a digestion of these lymph nodes. So when you do the digestion, you can detect the cells that are both in the parenchyma and the subcapsular sinus. Surprisingly, by flow cytometry, we didn't see a difference. So this means that the reduction of migration that we saw in the ear is not happening here. Probably it's being compensated with the intralymphatic crawling. But it could be that the cells, how can we know whether the cells are really stuck in the subcapsular sinus or not? What we did is like an immunofluorescence of the auricular lymph node of some sections. And here we can see in FITC and C11C the dendritic cells. In green, in red, we can see the subcapsular sinus, and the rest will be the parenchyma. This is done by segmentation. So we quantify all the cells that are in the parenchyma and the ones that are in the subcapsular sinus. What we saw is like here, what we can see is that the cells, the wild-type cells, in the wild-type mouse, are being accumulated in the subcapsular sinus, whereas we have less cells, dendritic cells in the subcapsular sinus and more in the parenchyma in the mutant. And this was done by calculating the percentage of FITC positive and C11C, who had more cells accumulated in the subcapsular sinus and more in the parenchyma in the uPA mutant. So in order to confirm this, what we did is there is an approach where if you don't digest the lymph nodes, okay, you show only the dendritic cells that are coming from the parenchyma, but not from the subcapsular sinus. And this is a way to confirm these observations. And indeed, first of all, we saw that when we digest the lymph nodes, we have more cells because we have the ones from the subcapsular sinus and the ones from the parenchyma. Okay, and there was no difference between them. But when we analyzed the migratory dendritic cells and the FITC dendritic cells, we saw that in the uPA, in this condition, analyzing only the parenchyma, there was an increase of migration of dendritic cells, confirming that the cells in the wild type are getting stuck in the lymph node, and they are migrating better in the uPA mutant. And this is probably because here we have less soluble than in the wild type. So these cells can bind better to the CCL21 that has a better binding it's been reported that the full length binds better to the CCR7 than the soluble one. So less soluble CCL21 is making the cells migrate faster and enter better into the lymphatic, into the lymph node. Whereas here, if there is less soluble, there is less directed migration towards the lymphatics. And finally, what we also saw is like, what is happening in inflammation? Because I didn't explain what is happening in the lymph node inflammation. And what we saw is that under inflammatory conditions, in inflammation, there is more cleavage of the full length in the lymph node that we didn't see in any of the other conditions. So there is reduced full length in the lymph node. And also we have more plasminogen and plasmin activity, which will explain why we have more, we have less CCL21 full length in this setting. And obviously here we will have more soluble 1 draining, but this is something we still need to explore what is the consequences under inflammation. So what could be the immunological benefit of such gradient adjustments? It could be that under inflammatory conditions, the CCL21 rapidly shifts to having more soluble in the ear, in the tissue. So we have more dendritic cell migration. And this dendritic cell and the inflammation migration is even more CCR7 dependent. So when there is an infection, only the ones which they really express CCR7 can mature and reach the lymph node. For example, other dendritic cells, such as the B-standard activated dendritic cells that can present these self-antigens, will not sense this gradient as good as the ones that they really have the pathogen-driven maturations. And I would like, of course, to acknowledge to Cornelia and also to all the members of the Halling group, but especially Nefeli and Philip and all of the collaborators that we had during this time. So I don't know, I'm sorry I tried to be a little bit fast, but I'm a little bit out of time.
No worries, Victor. And that was great. Thank you.
Yeah.
Folks, I'm conscious of time here, but if anyone has one question, maybe we could have Victor answer one. And
otherwise, always contact me by email, and I will be happy to.
Perfect.
Yeah.
I'll ask one just quickly then. Victor, so
Yeah.
I'm wondering with the plasmin activation, any specific cytokine within inflammation responsible for increasing activation of plasmin within LECs?
So, can you repeat, please? So what do you? So if
What
there is another
specific?
cytokine.
What's that, sir?
Sorry, I couldn't hear exactly what you asked.
So what specific cytokine do you know, if any, within inflammation is increasing the activation of plasmin?
Oh, okay. So obviously CCL21, but there is also another cytokine which is CCL19, which also is guiding migration from the lymphatic. So it could be that also CCL19 could have an effect, and maybe, but there is no reports as far as I know about Cliff CCL19.
No, sorry, I'm asking, all right, so for the activation of plasminogen to plasmin within LECs,
yeah.
what
So then
cytokine is driving, like interferon gamma, genotype?
So you have, so the activation, it could be either TPA or uPA. So TPA so needs to bind to fibrin in order to activate plasminogen into plasmin. But uPA just need to be bound to the uPAR. So it could be either both TPA or uPA, but we saw that the LECs, they express much more uPA and uPAR than TPA.
Got it. And then upstream of that, what causes uPA to go up within LECs?
So, I mean, you mean that the upregulation of uPA?
Yeah, exactly. Is it
Just
interferon
inflammation.
gamma?
It's been shown that, for example, TNF,
Got it.
LECs stimulated, they express. What we did is like, we observed that in mice under the CHS model, they express more uPA uPAR than the steady state ones. And we also did this in an in vitro setting with LECs by a TNF stimulation.
Beautiful. Okay. Thank you, Victor.
Basically, inflammation itself upregulates this, and it's when we saw more CCL21 cliff. Gotcha. Yeah.
Okay, awesome. All right, well, everyone, thank you for joining. This seminar will be recorded, and it has been recorded and will be uploaded to YouTube. And yeah, if any other questions, reach out to our speakers via social media or their emails.
Thank you, Dr. Nabil.
Thank you, Victor. Thank you, Xiangyue, and have a good rest of your days.
Ciao.